everyone. My name is Mariana Wolfner. I'm the president of the GSA, and I'm delighted to be here with you to introduce Ofer Rog and to welcome you all back to another lecture in our 2024 GSA Awards series. So this past March, Ofer was presented with the GSA Early Career Medal at the TAGC conference. This is a new award that honors an individual member of the society for their contributions to genetics at an early career level, which is defined as 7 years from starting an independent position. Ofer has been recognized for work in visualizing meiotic exchange between sisters, for exploring synaptonemal complex proteins and tracking single molecules. In addition to the science, which you'll be hearing about, Ofer's efforts to recruit and maintain a diverse student body at the University of Utah and to support LGBTQ plus students are very commendable and inspiring to many in the field. So Ofer is an associate professor of biological sciences at the University of Utah. He studies chromosome biology, meiosis, synaptonemal complex, worms, yeast, and microscopy. The Rog Lab studies chromosome-wide regulation, which is key for almost all aspects of biology and health. Their website explains that during sexual reproduction, regulated interactions between chromosomes are essential for chromosome segregation and generating genetic diversity. At the same time, cancers capitalize on the disruption of chromosome-wide regulation and the ensuing loss of genome integrity and aberrant gene expression. But many of the organizing principles and molecular mechanisms that underlie this chromosome-wide regulation remain a mystery, and that's what the Rog Lab studies. In Ofer's seminar, you'll hear 2 unpublished stories. The first one documents the unexpected demixing of sister chromatids during meiotic prophase and the mechanisms for this. The second one describes a new genomic technique that is lab developed to characterize large-scale chromatin organization and then applied it to meiotic chromosomes. Ofer co-founded the University of Utah's LGBTQ plus STEM group, which supports LGBTQ plus students and invites speakers to discuss their science, their activism, and to share personal stories to build community. He's mentored and advised first-year graduate students for many years and has fought actively to improve mental health insurance coverage at the university as part of the academic senate. He's also created intensive fellowship writing course for graduate students to address an unmet need for science writing skills. So in a moment, I'm going to turn this over to Ofer, but before that, 2 quick housekeeping items. Secondly, I just want to echo Jessica's comment that this, like all GSA events, is covered by a code of conduct, and the link is in the chat. The second is to ask you to put your questions for Ofer in the chat. We'll get to as many of them as possible in the Q&A portion. I will call on you, and then you can unmute and ask your question. But for anyone who wants to ask a question anonymously, please send it directly to Jessica, and she'll ask it for you during the Q&A. So with that, I'm delighted to hand over the Zoom to Ofer. We are really looking forward to your talk.
Thank you again for this introduction and for this opportunity to talk today. I've used my acceptance speech at the GATC a couple of months ago to thank my many friends and mentors along the way, which are definitely none of this would have been possible without them. And for today's talk, it's really exciting that I get a chance to talk about the work of people in my lab, which is really the kind of the reason I'm here and we're all here. So going right in, my lab is focused on studying chromosomes, and I feel chromosomes are potentially a bit misleading as an object of study, just because they are, as we all know, made out of DNA. But even though they are made out of DNA, because they're so much bigger than naked or bigger than the base pair events at the double helix, many times the regulatory processes that are going on to allow them to function are not trivially deduced from events that occur at the base pair level. And a corollary to that is that the techniques that we need to use to probe these processes are many times different from events that are used, or the techniques that are used to probe events at the base pair level. And I hope that's going to become apparent during the talk. The biological process that my lab studied is meiosis. This is the specialized cell division that produces gametes like sperm or egg. The main goal of meiosis is to put exactly one copy of each of the two parental chromosomes or homologs into the gametes. Upon fertilization, two gametes merge to restore this kind of two-copy situation in the zygote and later on in all the cells in the body. The way meiosis achieves this feat is somewhat non-trivial. So upon entry to meiosis, the cell deliberately shatters its genome into many small pieces by introducing many breaks. And it is the process of repairing these breaks that generates exchanges or crossovers between the parental chromosomes. And these crossovers are essential to allow the chromosomes to segregate correctly into the gametes. Now, for quite a while now, first in my postdoc with Abby Dernberg and then in my own lab, we've been focused on studying the synaptonymal complex, which is a conserved interface that assembles between the parental chromosomes. It has this very stereotypic appearance in EM as a ladder. And we've published a lot, and we're still working a lot on understanding the mechanism by which the synaptonymal complex brings chromosomes together and regulate exchanges between them. However, today, this was with a heavy heart, I decided not to talk about the SC, just to have a chance to expose some new work in the lab that is actually related to chromatin and how is it organized to allow meiotic processes to happen. So as Mariana introduced, the two stories I want to talk about today are focused on sister chromatids and how they are positioned relative to one another, and chromatin organization more broadly. I like this particular choice of two topics also because they highlight the different techniques that we use in the lab, the sort of very different approaches that we use to tackle these problems. So I'll begin by telling you about the work that Antonia, a very talented graduate student, has been doing in the lab. So just to get everybody on the same page, sister chromatids are the two identical products of DNA replication. And during the cell cycle, this is in every eukaryotic cell. This is not specific to meiosis in any way. The cell kind of cycles between states when there is one copy of each chromosomes, and after DNA replications, we have two copies of each chromosomes, and these are called sister chromatids. And these are, as you can imagine, impact many cellular and chromosomal processes, but because sister chromatids are identical, actually studying how they interact with each other and how they're organized is very non-trivial. This question of interactions between sister chromatids is particularly important during meiosis. And the reason for that is that when the kind of events of meiosis that I told you about a couple of slides ago occur after DNA replication. What this means is that we have four very similar DNA molecules, two parental chromosomes, homologs, each made out of two identical sisters. What this means is that if we are introducing a break now, this break can be repaired in two different ways. It can use the homolog as a repair template. This kind of repair has a chance to yield a crossover, which again are essential for formation of gametes. But this repair might just as well get, this break might get repaired using the sister chromatid. And in this case, we won't make a crossover. And if we fail to make a crossover on the chromosome, we would get aneuploidy and sterility. So it's perhaps not surprising that repair in meiosis is heavily biased toward the homolog. However, the mechanism by which this occurred, the mechanism that sort of allows the chromosomes to know which is the sister and which is the homolog, is very much unknown. So we've been interested in this question for quite a while. David Almanzar, a previous graduate student in the lab, developed a technique to specifically label one of the two sisters. This uses substitution of thymidine to EDU. And if you time the timing of introduction of EDU correctly, you can get a situation where you get two sisters. They're still going to be have the same identical genetic information, they're exactly the same sequence, but only one of them would have EDU, so we can distinguish the two sisters cytologically. This works beautifully. So what you can see here, this is a pair of chromosomes at the end of meiosis. So this is a chromosome just before it's about to divide. You can see here that one of the two sisters is labeled and the other one is unlabeled. And I hope you can also appreciate that in this particular case, these labeled sisters remain nicely confined in this kind of quadrant of this pair of bivalents. And this is in contrast to conditions like that. So this is a particular chromosome here where we have one labeled sister and it starts off in this bottom right quadrant, but then it sort of moves over, leaks over to the other side. So this is a case where the two sisters exchange information between them. And what quickly became apparent to David is that non-exchange chromatids vastly outnumber exchange chromatids. And we've spent quite a bit of time characterizing some of the mechanisms that generate that strong preference. This work looks at the products of DNA repair, sort of kind of what happened in meiosis that allows kind of how we look at the chromosomes at the end of meiosis and sort of deduce what happened early on in meiosis. So if you look at it cytologically, all this work was done in C. elegans. This is the C. elegans gonad. In here, what you see here, these are many germ nuclei that are going through meiosis as they move in the gonad. And the chromosomes that David looked at were here right before, at the very end of meiosis, right before the meiotic divisions. What Antonia wanted to study was how are sisters organized at the time when repair is taking place, at the time when you actually need to make the choice of how to repair breaks. So this stage, we refer to it as meiotic prophase, but I should stress that at this stage, the chromosomes are very much resembling interphase chromatin in the sense that they are very extensively transcribed, and obviously they're undergoing break formation and repair. So in many ways, they resemble the interphase chromosomes. In that stage, we know that the two homologs are positioned kind of right next to each other, side by side, with the synaptonymal complex mentioned before in between them. So here it's in pink. So we have the two homologs here, but how the sisters are organized, we don't really know. So this is what Antonia wanted to figure out. She tweaked David's labeling protocol to enable her to look at this stage of meiosis. So again, to remind you, we have two sisters, one of each pairs of homologs is labeled with EDU. The images that she got were very revealing to us. So what you see here in magenta is chromatin. So that's a general label for DNA. It labels both sisters. And then in green, we have labeling of one of the sisters. And what we see is this kind of patchwork of regions where we are seeing one sisters and region where we're seeing the other sister. So this is sort of a diagram to highlight what we see. And so what this means is that these clusters that are hundreds of nanometer in size must include multiple chromatin loops from one of the sisters, whereas these purple regions here must include loops from the other sister. Just to drive this point home, what this entails is that the distance between the two homologs, sort of between the two sides of the SC, is on the same size scale as the distance between the two sisters. So between the green, the labeled, and the non-labeled sister here. So this was quite surprising to us, and I'm going to get back to the implications of that at the end. These images, as you can imagine, are somewhat challenging to quantify and analyze. Antonia is still working on that, but she resorted to using a a simplified condition that allowed her that made a lot of these experiments simpler. This is using a mutant where one of the chromosomes does not pair. What this means is that rather than having 4 sisters side by side, we only have 2, in this case, the 2 sisters of the X chromosome. In this particular case, we can very nicely see the separation. This is even on focal microscopy. Again, this is both sisters labeled in DNA and only one of the sisters labeled with EDU. The second sister here is unlabeled. Again, just to drive home what is happening here, when these 2 DNA sisters left the replication fork, you can think of them as sort of long noodles that are tethered at multiple sites along their length. In order to get to this type of arrangement, what needs to happen is that all of the sister on top, the blue sister here, must form loops facing in one direction, and all this sister in the bottom needs to form loops facing in the other direction to kind of generate this type of arrangement. So how does that happen is really the core of what we'd like to understand. Antonia developed a volumetric assay to quantify these kind of images. This is shown here. What you see here is the volume covered by DAPI, so both sisters, a volume covered by EDU here in green. This is one of the sister. The ratio between EDU and DNA here is 50%, which is, again, the maximum separation that's possible. I'm going to refer to it in the text as spatial differentiation. So this is the kind of the demixing of the sisters. Antonia did a lot of controls for her labeling and her quantifications. I'm going to show you a few of them. This is one here. In this case, she's comparing situation when we label one of the sisters versus conditions where we label both sisters. As you'd expect, in the both sister labeling configuration, we see close to 100% overlap versus the 50% overlap. In the single sister labeling, there's a few more controls here. She's looking at a different unpaired chromosome, again, to make sure this is not something specific to the X. We get similar level of different spatial differentiation. And then this was important for us. The stage that we look here at the chromosomes is quite a few hours after replication. So maybe 10, 12 hours after replication. Meiotic prophase in worms is quite long. The condition here, we're looking at cells much closer to the time of replication. We can't look at them right after replication, but this is probably 10 hours sooner than this. Still, we're getting very similar level of spatial differentiation. So the differentiation is not merely a result of just spending a lot of time prior to replication. The next step, so we're convinced we're seeing a real differentiation here. Antonia spent a lot of time trying to figure out some of the molecular mechanisms that regulate this. And I'm going to show you one key result from her finding. Her candidate analysis pointed her toward a role of cohesins. These are conserved ring-shaped molecules that play multiple roles in organizing chromosomes. In meiosis, 2 of their key roles are tethering the 2 sisters together. So then here, this is performed by this orange subcomplex. And then another role that they do is they extrude these, they act as motors to extrude these chromatin loops. And this is done by this subcomplex here in purple. To do this loop extrusion activity, cohesin needs to be loaded onto chromatin, and this is performed by this factor SEC2. Now, as you can imagine, we can't just look at null mutants of cohesin to analyze this process. These chromosomes are going to be completely disorganized. So Antonia utilized the conditional Degron system, the AID system, and then she optimized the time of the amount on auxin, sort of the length of time in which SEC2 is being degraded. You can see here that after 10 hours on auxin, SEC2 is gone. But then she could use the unique organization of the C. elegans gonad to be able to analyze the situation here. So what you have here on the very left, you have nuclei that have entered meiosis, went through DNA replication, all without cohesins. These nuclei, the chromatin there is very disorganized. However, here on the middle and the right, we have nuclei that entered meiosis before they were placed on auxin. So the reduction in auxin does not completely destabilize chromosome structure. This is kind of highlighted here. We still see these kind of nice threads of SC between the chromosomes. These chromosomes, she could now sort of compare apples to apples with a wild type setting. And this is what she's done here. This is a gallery of a few of these chromosomes. And I hope you can appreciate that the green signal looks much more diffuse. In these, we don't get this kind of half and half separation we get in the wild type setting. And this is quantified here. So what we see here is that this activity, this loop extrusion activity of cohesins is necessary for this differentiation of the sisters. So unfortunately, I don't have time to show you all of our data, so I'm going to summarize now, although I'm happy to take more questions on this. So what I've shown you in this part is that single sister labeling revealed that these sisters are spatially demixed, spatially differentiated or demixed early in meiotic prophase. This does not require a long time in prophase, and I didn't show you this, but it also does not require the activity of condensins. However, it does require this loop extrusion activity of cohesins. Going back to this questions about how does the cell identify or is able to effectively identify the homolog from the sister to send repair into this pathway. A lot of the models that have been proposed to solve this problem relied on the idea that the sister is close, but the homolog is far away. That was been the sort of working idea of the model. But our finding is that the sister and the homolog are probably, spatially at least, they are in the same distance, suggests that the cell would not be able to rely on this cue in order to differentiate the sister and the homolog. And still, how the cell can efficiently do that is not, I think it's still a mystery to me and something we are thinking a lot about. So I'm going to end with this story now and move on to the second story I wanted to share with you. And this is the new genomic technique that QA, a graduate student in the lab, has developed. QA has been working on this for quite a few years now, and he's graduating next week, so his defense is in exactly a week. We've posted some of this a couple of weeks ago, so I urge you to take a look at this work. So the question QA was asking was quite related to what Antonia was asking. So again, we're thinking about long noodles of DNA that enter meiosis. When they enter meiosis, they adopt this array of loop structures that are anchored at its base to a proteinaceous structure called the axis that's made of cohesins and other proteins. And this organization as an array of loops is necessary to allow the chromosomes to interact with one another. The way this has commonly been studied is through the genomic techniques like ChIP-seq. And the way this works is that you take meiotic cells, you chemically cross-link DNA and protein, you shear the DNA into small pieces, you identify the pieces of DNA that bind your protein of interest, say cohesin, and then you sequence them, and then you know which sites of the genome are preferentially associated with cohesins. This has been very powerful, but there's some questions that, because of the nature of this approach, it's very difficult to answer or impossible. For example, giving you an example of such question, if you want to ask how is cohesin association on one site affects cohesin association on a faraway site on the same chromosome, that kind of question is impossible to answer because you've sheared the DNA into small pieces, you effectively erased all of that information. So in order to address this, QA decided to develop a new technique. We call this check, please. And I'll tell you sort of like the general principle of it. So we developed this in budding yeast. Although, as I'll hope to convince you, there's nothing in this approach that prevents it from being moved on to other model organisms. The logic is as follows. We start by tethering a DNA methyltransferase to a protein of interest, in this case, cohesin. And this DNA methyltransferase is going to methylate DNA in proximity to where cohesin is. This this idea, we got the inspiration for it from beautiful work from Steve Henikoff's lab. The next step is different. We take DNA from these meiotic cells and subject it to long-read sequencing with nanopores. For those of you who are not familiar, the way this works is that you thread very long pieces of DNA, so they could be hundreds of kb in length. You thread DNA through a protein pore, and by detecting the current from on either side of this membrane, you can deduce the sequence of the DNA and also any kind of modification that of any base in the DNA. What this allows you to do is to identify the kind of clusters of methylation, for example. And when you see a cluster of methylation in your reads, you know that that region of DNA has been in contact with cohesin. So it's allows you sort of to decode how the chromosomes was organized. This took quite a while to get set up, but this is the kind of data that QA was able to get from these type of sequencing. So what you're seeing here are a sample of about 10 or 15 individual reads that span this 20 kb region on chromosome 3. Each one of these dots is a GATC site. And then in blue, we have sites that are unmethylated, and in yellow, methylated sites. What I hope you can appreciate is that first, some sites tend to be much more methylated than some other sites. We knew that from before. You can also appreciate that every single read is unique. This is, again, inherently a single molecule approach. We wanted to validate it, and we did all sorts of things. I'm going to show you a few of them. For example, here we are looking at the accumulation of methylation over time. This is the way this experiment is set up. You are inducing a meiotic culture into meiosis, and then the more the cells are in meiosis, you're going to get more and more methylation. Budding yeast doesn't have a demethylase to remove it, so it basically just accumulates over time. And this is indeed what we see. So you can see between 3 and 4 hours, and between 4 and 5 hours, we see accumulation of methylation. You can also see that we see enrichment of methylation around centromeres. This is known for cohesin, so this is expected. And we also, in general, see this kind of seesaw pattern. And this seesaw pattern, what it tells us is that there's some sites that tend to be associated with cohesins. These are sites that are thought to be close to the axis. And then we have sites that are low in methylation, presumably associating less with cohesins and away from the axis. So this is kind of how you generate these loops, this pattern of loops. We also wanted to directly compare this data to ChIP-seq data. Before I show you the data, I'm just going to remind you that these techniques detect very different things or related things, but are not exactly the same. ChIP-seq gives us a snapshot of where is cohesin at the moment where you stop the experiment, whereas checkplates give us a historical view of where cohesin has been throughout the time it has been expressed. The patterns that we get are similar, but they're not identical, which is what we thought. We get, again, a lot of the peaks are overlapping, but we do get some regions where, for example, we see a check please signal, but we don't see a ChIP-seq signal. And we don't know about the nature of all of them, but at least for some of them, we hypothesize that these, for example, are sites where cohesin is loaded, but has moved away from that. So that's going to give us a positive check please signal because cohesin has been there at some point. So, so far, I've shown you how we can recapitulate known data, but the real power of this approach is again, that we have these long reads and individual, that we can compare individual reads. So I'm going to show you two specific advantages of things we can get from this data that we wouldn't have been able to get from a ChIP-seq data. And the first relate to our ability to analyze single reads. So in here, I'm showing you here on the left, three extreme hypothetical examples of reads, and we're looking here at two GATC sites. In each one of these groups, we have 50% methylation on each of these sites, but the arrangements of methylations relative to one another is different. So for example, here we see a case of complete correlation between the two methylation, complete anti-correlation or randomness, and we quantify this by this measure, which is the log of the coefficient of coincidence. So this just gives us a positive value for this type of scenario and a negative value for this sort of repulsive or anti-correlated scenario, with 0 being just random independent events. When we analyze this data genome-wide, so what we're seeing here, this is this log COC value, and on the x-axis, you see the distance between GATC sites. What became very clear is that we see positive GATC site at effectively all the distances that we look to, from very close site to sites that are 10 kbs shown here, but this keeps going for 20 and 30 kb away. So we get consistent positive correlation between GATC sites. So if we have a GATC site on one read, we're going to have another GATC site nearby, but actually far away. This correlation is lost in the shuffled read. So this is just a control for ourselves where we shuffled methylation values between the different reads. And that obviously, as expected, we get 0 here. So this was observation was puzzling for us for a while. And based on several lines of data, our hypothesis is that what we're seeing here is the consequence of movement of cohesins along DNA. What that's going to do is that as cohesin moves, it's going to leave methylation marks along the same DNA molecule, resulting in this positive correlation. I'll show you one piece of evidence for this fact. For this, I'm going to show you a variant of checklists that KOA developed with Ethan, a rotation student. In here, we're expressing REC-A tagged with GFP. So this is different. This is not REC-A tagged with DAM, but it is tagged with GFP. We induce these cells into meiosis, and then we isolate nuclei from these cells. So by isolating nuclei, we are stopping all of the energy-dependent processes, for example, transcription or translocation of cohesins on DNA. So this is a motor-dependent activity. Then we take these nuclei and we incubate them with a recombinant protein that includes a GFP binding protein that is tethered to DAM. This is going to go into the nuclei, interact with cohesins, and methylate nearby DNA, and then we can take the DNA out and sequence it as before. I should say that this approach of methylating isolated chromatin has been used by several different labs that produce this kind of alphabet soup of different techniques here that's been published over the last few years. Not unexpectedly, we get pretty similar profiles using this nuclei isolation method versus in vivo. You can see the overall profile is quite similar. Again, we see some interesting differences that I'm happy to expand on in the question if people have them, but overall the profile is quite similar. What was striking to us is that even though the profile is quite similar, if we look at the correlation between different sites, it is completely lost. So we're basically getting 0 random correlation on all different sites. So methylation becomes random. Again, this is consistent with the idea that the correlation in methylation require the active movement of cohesin on DNA. So this is what we think is happening here. This data also gave us another kind of very interesting observation that also serves as a kind of nice control for this. So what you're seeing here is bind methylation by by distance. But if we look at this very short range correlations here, what we see is this striking oscillating pattern that the periodicity of this is 165 base pairs. So this periodicity is apparent in the in vivo setting, but it's actually much more apparent in the nuclei isolation. Again, just to highlight, overall, we see correlation that's 0, but the amplitude is actually quite high over here. And this amplitude, the periodicity exactly matches the distance between nucleosomes in vivo in budding yeast. So what we speculate is happening here is that cohesin somehow is methylating nearby nucleosomes. And because these nucleosomes are stacked with each other, we're methylating the same region on OH nucleosomes. There's several other explanations for this, but that's something we're trying to tease apart now. The final thing I want to show you is another thing we can do with the data that we obtain with Check Please. And this is, to do that, I'll introduce you to the rDNA locus in budding yeast. So the way rDNA is encoded is in a giant locus on one chromosome, where we have 100 to 200 tandem repeats of a perfect 9.1 kb unit. It's hard to sort of like get how big that is. So this is a single locus that harbors 10 to 20% of the budding yeast genome. Because of the size of these repeats and because they're identical, a technique like ChIP-seq cannot distinguish between them. So you can just pull all your reads to a one unit, but you can't tell how repeats are different from each other. Because in our technique, we have very long reads, we can map them to unique sequences that are just outside here. So this is what QA has done. He mapped repeats to these unique genes here, and then was able to see methylation pattern in the rDNA. I don't have time to show you all the analysis, but just, for example, one fact of the data that we can get here, we see consistently higher methylation inside the rDNA repeats versus outside, which tells us that, or suggests that there is higher occupancy of cohesins in the rDNA versus the outside of the rDNA. So I'm going to end with the data here. Just to summarize, I hope I convince you that Check Please has great potential to elucidate regulation of events in the chromosome on a large scale. It can record dynamic events on chromatin, and it can allow mapping or extension of protein binding information even into highly repetitive regions. These kind of advantages sort of, I think, by themselves suggest the kind of questions we're trying to answer right now that QA is working on. One of them is looking at different dynamics event in the cell, for example, homology search or transcriptional activation. The other thing we're trying to do with it is looking at organization of heterochromatinized region. These tend to be highly repetitive, very large, and not easily addressed by other techniques. For some of these, we might have to move to a different model organisms that has more complex centromeres, for example. But as I mentioned, that shouldn't be, that should be quite easy with this approach. So again, I want to thank my lab and apologize to those lab members whose work I wasn't able to share with you today. They are a really great group of scientists, and they make my daily life really a joy. Also, again, for the GSA for this award and for this funding. So thank you.
Thank you very much, Ofer. That was really interesting and thought-provoking. I wanted to open up the floor for questions. We have a question from Charles Laird in the chat. So Charles, if you want to unmute yourself and turn on your camera, you can go ahead and ask. And okay. Please, anyone else with questions, please put them in the chat. Thank you.
Yeah, thank you very much. This EDU method looks like it might be able to make some inroads into that very old paper by Hoda and Stern on meiotic DNA replication, unexpectedly late. And this also could tie in with Woodrow's work on late somatic replication, which appears to occur after major condensation has already occurred. Have you seen anything or approach those questions?
I would say the one thing that we do see a lot, and this is observation, the original observation was not by us, was made by Johan Engelbrecht, was about and looking at our images, you might have asked yourself, how do we get just the X chromosome to be labeled? And the reason for that is that the X chromosome does replicate much later than the autosomes in worms, which has been convenient for us sometimes if we want to time our experiments right. The resolution of our ability to, or what we've done so far in trying to resolve timing between early and late replication in the same chromosome has been quite limited, at least at the level of cytology. We do occasionally see cells that, would be consistent with, that we managed to get the EDU there, just kind of at the very end of the S phase. I would say that I don't know how much cytology is the way to go there. Probably a sequencing-based method is going to give us better resolution, but we are thinking about that. Yeah.
Yeah, and maybe with cells with larger chromosomes, you'll get a better discriminant.
Yeah. I mean, I wasn't harping on that point too much, but I should say that the cytology that we do get with worms is quite exceptional, and definitely superior to a lot of organisms. All the images I've shown you, I should probably have stressed that more. These are all whole mount, so these are not spread chromosomes, and just the fact that one could see even chromosomes, not even talking about sisters, but whole chromosomes separated from one another in a whole mount situation where chromosomes are, again, not mitotic, is pretty unique, and in many ways set us up for the ability to see single sisters.
It'll be very interesting to see some of this applied to chromosomes that have discrete centromeres instead of the diffuse ones. Do you have plans or data on that?
I mean, you're talking about the cytological work, just to clarify.
The diffuse centromeres, which I understand C. elegans has.
Yeah, C. elegans they're holocentric and they do have diffuse centromeres. Yeah, we haven't introduced the EDU system to any other organisms, but I do agree it would be great to do, and I don't think there's any inherent limitation to doing that.
Yeah, it's terrific inroads. Thanks very much.
Thank you.
And we have a question from David Almanzar. Can you unmute yourself, please?
Hi, Ofer. So great to see how this project has progressed. I was wondering whether you've investigated whether the lack of sister separation influences the total number of sister exchanges.
No, that I think would be that's definitely that's a great question. Thank you. And we definitely are thinking about that. The other side of this question, which I assume you would also think about, is how does for example, lack of sister separation would affect the homolog bias, which I think is a key future question for us to figure out. So we're definitely thinking about that.
Thank you. The next question is from Anne Villeneuve. Can you please unmute yourself?
Hey, Ofer, that was really good. And the question I'm thinking about is like these beautiful findings that you have from Antonio's data. How are you thinking about those in relationship to some of the nice data from Alex showing the stoichiometry and spacing of the different cohesin complexes and the asymmetry of loops?
Great question, and this is a great reminder to I should have noted Alex's paper here. So
I wasn't trying to push on that. I was more
no. I am. So I would say that a slide I did take out because I didn't want to go too much over time was a slide where I have two movies from simulation from Leonard Mirny's work. And what they showed is that entropic repulsion between DNA polymers is enough to separate them, but not if you have cross-link between them. So we do know, obviously, there are cross-links. And one of the things that we are trying to do now is trying to see if we can sort of dial up or down the number of cross-links along the chromosomes and trying to see how that might affect this, the ability of the sisters to separate from one another. So I think the kind of data that Alex found about the number of molecules, at least, which we can hopefully translate directly to the number of like tethers between the sisters, how would that affect, and whether we can sort of tune it up and down and see how that affects sister demixing. That's definitely again, I wish I would have had more time and then I would have said it, but thank you for the question. Yes.
Thank you. The next question is from Boris Leibovich.
Yes, my question concerns working very often with CTCF protein in the complexes. Do you think to engage also analysis of CTCF for the processes which you observe?
So thank you. The budding yeast does not have CTCF, and neither does worm. I hope I'm not mistaken here. But so the exact way cohesins are positioned in different kind of eukaryotic lineages seem to sort of diverged a bit, and it's not always the same in budding yeast, at least based on a lot of work that again was in fact deduced from ChIP-seq among other methods, show that in budding yeast the position of cohesins is mostly by transcription. So transcription seems to position where cohesins are. I think in worms that is much less well understood. We don't have a clear kind of CDCF-like molecule that is able to allow us to predict where cohesins are going to be. So we don't have an easy way to do that. We are trying to look at, Antonia definitely is examining other regulators of cohesins to check their effect on sister demixing.
Thank you.
Thank you. We have an anonymous question. I'm just going to read it. It says, "Thank you for your beautiful talk. I have a question about meiosis. What are the known key factors that are required to initiate meiosis?"
Yeah, a great question. And not always known. So in budding yeast, that's it's mostly about nutrition. So the way you induce meiosis is you take diploid cells, obviously, and put them in a starving condition. So stress is what prompts them to do meiosis. In worms and other organisms, this is more of people think of it as a kind of a stem cell niche and differentiation process. So at least in worms, again, if you remember the gonad I showed before, the part on the left is acting as a stem cell niche, and cells are dividing again, nuclei are dividing vegetatively there. The nuclei that escape that niche, then they kind of enter meiosis. So yeah, I hope that was answering your question. Yeah.
Well, I also have a question of my own, which is pretty naive, but if the sisters are about the same distance from each other as the homologs are from each other, do you get an equal number of corrections from sisters as from the homologs, or is it biased? And if it's biased, do you know why?
No, I think that's I would say that the observation, let's say in worms, where Anne's lab actually has done some of the beautiful work on that, it seems that if you make a single break, it's going to invariably use the homolog. So it seems to know very well where to go. The exact extent or the exact extent of this bias, again, this is not a very easy measurement to do. but it does seem to be conserved, not in direction, but not necessarily in exact number. Again, so I think to answer your question, we think that the repair is heavily biased toward the homolog. The situation might become a bit more murky when you have multiple breaks, but they do seem to know, quote unquote, which one is the homolog. How they do that, I don't know. I think, again, physical proximity based on Antonia's data suggests to me that by itself, at least, cannot be responsible for this very strong bias. But whether there are other factors, for example, other structural factors that are involved there, that's a big mystery to me. I think Nancy Kleckner and others have worked in yeast suggesting cohesins are responsible for that bias, at least partially, but I think it's still a very active area of investigation.
Thank you. Beautiful work and talk, just blew us all away, and you answered all the questions. So thank you so much for your talk.
Thank you all for coming.
And congratulations again on the award. And thank you everyone else for coming to listen and ask questions at this talk.